Welcome back, folks. This is Chapter 15 on Recombinant DNA Technology and its applications. Some of the material in this chapter has already been covered in other chapters, so that'll make it easy and a revision tool. The authors have divided the chapter into 4 parts. Each part deals with a certain aspect of the technology. Recombinant DNA technology has a definition, and the definition varies depending on who you ask. According to our authors, recombinant DNA technology is just a set of techniques that are used for magnifying, known as amplifying, maintaining, and manipulating DNA sequences both in vitro, that means in a test tube, and in vivo, inside an organism. Because the DNA of even the smallest organism is pretty large, the challenge is to identify the specific region of DNA that one wishes to manipulate and isolate that in vitro. The goal until more recent technology kicked in was to allow researchers to manage the genome in tiny chunks that could then be analyzed and then, if necessary, reassembled to provide a better view of how genes and genomes are functional and arranged. Traditional recombinant DNA technology was used to first break up the DNA, called fragmentation, into manageable sizes, and then to isolate the pieces of that DNA that the scientist was interested in. Then to create multiple copies of those isolated fragments so that enough research could be performed. Those fragments are then placed inside more manageable DNA molecules that may belong to an organism in which they can be grown, and then to determine, if required, the exact sequence of the fragments obtained. These researchers could go on then to identify fragments of DNA containing complementary sequences. And once those were identified, introduce specific DNA molecules back into living organisms, and then determine the phenotypic effects of those isolated DNA fragments. Some of the original tools used by molecular biologists and geneticists from the beginning have been restriction enzymes. Restriction enzymes are simply, in the vast majority of cases, proteins made by bacteria. And those proteins are designed to intercept foreign DNA that has entered the cell. And they do that by cutting across 2 strands of DNA, the double helix, and therefore fragmenting the DNA. The restriction enzymes were first isolated in bacterial cells and since have been found in archaea and some other species. Their natural purpose is to act as an immune system, protecting the host cell from viral infections. Luckily for the host cell, these same systems have a method of methylating the host DNA so it doesn't get attacked by its own restriction enzyme. So as we know from previous lectures, methylation is simply tagging the backbone of the DNA with methyl groups. And once those methyl groups are in place, the restriction enzyme cannot recognize that region of the DNA. The problem is, if a virus learns how to do this, then the next time it infects the same cell, it may also have methylated the same recognition points on its DNA. Many hundreds of restriction enzymes can be purchased from supply companies, and each restriction enzyme is normally named after the species in which it was first identified. And if that species has more than one restriction enzyme, we just add on a number to the end. The mode of action of restriction enzymes can be classified into 2 buckets. Those that generate sticky ends once they cut, and those that generate blunt ends. Let's look at sticky ends first. Sticky ends is simply a term used to identify a staggered cut. So Echo R1 was recovered from E. coli, and it was the first restriction enzyme to be isolated. It recognizes this pattern. So this is the profile that the enzyme looks for on any piece of DNA. And you can see the sequence read forwards is the same as the sequence read backwards on the bottom strands. GAATTC, GAATTC. So this is called a palindromic sequence. Whether you approach it from the top or the bottom, you will still recognize this area. This is a very important term in biology. Regardless, the restriction enzyme that we're looking at here, EcoR1, recognizes this sequence and no other, and it cuts the DNA between the G and the first A on both strands. And therefore, it leaves this 4-base overhang on each half of the cut. And this is called a sticky end. Why? Because these two A's and these two T's still have their hydrogen bonds available to bind with something that comes past. If another compatible molecule comes by, such as the same end that's been cut, then it has the potential to sit down next to its partner and form some type of complementary base pairing. But it doesn't have to be the fragment. It could be another fragment, or it could be the same fragment but upside down. So sticky ends can be useful and also detrimental at the same time. Therefore, techniques have been developed to ensure that the scientist gets what they are seeking. Other restriction enzymes naturally cut across both strands of DNA at the same location, therefore creating blunt ends, so they have no stagger or overhang. These types of DNA ends can be manipulated further using a variety of techniques which have been developed over the decades. With respect to restriction enzymes in general, they recognize between 4 and 8 base pairs of DNA. So EcoR1 recognizes 6 base pairs of DNA. So it's called a 6 cutter. So a 4 cutter and an 8 cutter, a 6 cutter. There's also 5 cutters and 7 cutters, but the range is from 4 to 8. The use of these restriction enzyme tools by scientists depends on what they want to achieve. If they want to cut the DNA frequently, they would use a 4 cutter. If they want to cut their DNA infrequently, they will use an 8 cutter. The size of the target DNA also computes into this calculation. One branch of investigation with restriction enzymes is that scientists have been able to map DNA sequences based on restriction maps. So these are not genetic maps, they're not physical maps, they're called restriction maps because they rely on overlapping fragments of DNA that are being cut by various restriction enzymes. And understanding that is the purpose of the next slide. The example that we are taking is Lambda genome. Lambda is a bacteriophage that attacks bacteria like E. coli. It has a relatively small genome of about 50,000 base pairs. If the only information one had was the size of the fragments obtained, then one could deduce where the restriction enzymes are cutting. So in this particular experiment, we're going to cut Lambda genome with APA1. And APA1 happens to cut once, somewhere along the linear chromosome of Lambda. And it generates 2 fragments. That's all we know. The first fragment is about 10,000 base pairs, and the second fragment is about 40,000 base pairs. So that tells us that the cut must be 10,000 base pairs from either side. So this just gives you one example. Then when you use a second restriction enzyme, ZO1, ZO1 will cut also once, and it generates a fragment that's 15,000 base pairs and about 33,000 base pairs. The question is now, which side does ZO1 cut and which side does APA1 cut relative to the beginning and the end of the chromosome? Remember, the chromosome could be in either orientation. And the answer to that is obtained by doing a double digest, where you add the 2 enzymes to the same reaction mixture and you allow the reaction to proceed. Now, if APA1 cuts on one side and ZO1 cuts on the other, then you will get 3 fragments, 15, 10, and about 23,000. If both enzymes cut near the same end, then you get a different set of fragments, with the smaller fragment being 5,000 bases, and then 10, and then a 33. So by combining different restriction enzymes together, you can generate maps. And here we see that the restriction mapping of Lambda was completed by using a number of different enzymes, in this case 4. So we have HIN-D3, ZO1, XBAR1, and APA1. The restriction profile generated from this electrophoresis gel was used to generate this restriction map. So this came first, and from putting all these pieces together like a detective, the scientists were able to generate this map. Experimentation with restriction enzymes is not just restricted to small genomes. Any genome is fair game for digestion. But of course, the more DNA you have, the more fragments you'll generate, and the more variety of fragments will exist. In a large genome, like that of a human being, where all the genetic material in the chromosome is digested, say with Echo R1, you will generate about 750,000 different pieces. And those pieces will be of all different sizes. And when you run them out on a gel, you'll get a smear. And a smear indicates that you have lots of different size fragments. Figure 15.2 shows a restriction digest of the genomic DNA of a moss. A moss is a plant. And you can see here, by using different restriction enzymes, ALU1, HIN-D3, ECHO-R1, BEAGLE2, BAMH1, and NOT1, you get different profiles, because each enzyme recognizes and cuts, that's where the asterisk is, recognizes a different length and different sequence of DNA, with ALU1 cutting more frequently because it's a 4 cutter and it generates smaller fragments of DNA on average, whereas NOT1 is an 8 cutter and it generates larger fragments. Reading these gels is important. This is a marker lane. This contains fragments of DNA of known size, which can be purchased from companies. And in the remaining 6 lanes, we have each one of these enzymes. So this fragment here, 23,000 base pairs, always travels relatively slower than the smaller fragments. And as you get smaller and smaller in your fragment size, they tend to drift further and further down the gel in the same time period as the smaller band. The DNA was loaded here in the wells and then subject to movement down the gel. Incidentally, as it says, in these plant species, the chloroplasts, of which there are many per cell, light up as discrete bands because those small chromosomal, circular chromosomal entities inside chloroplasts and mitochondria are going to digest to the same size. Once you have broken your DNA into fragments using restriction enzymes, what do you do next? Well, that depends. There's a multitude of avenues available these days. One of them is to clone your fragments. Cloning entails taking these small pieces and placing them inside vectors. Vectors are simply manageable pieces of DNA that can be placed inside living organisms and then grown into large quantities. And when you do that, you consider that an amplification process. When you have a single plasmid, for instance, the vector, holding one of your fragments, and that plasmid is present inside a single bacterial cell, then that cell can be grown, amplified into a large number of cells by just simply letting it grow overnight. Therefore, every single descendant of that one cell will contain the same plasmid with the same insert, and that is then called a clone. This is easier said than done sometimes. So the steps to molecular cloning are illustrated over the next few slides. So the vector, the piece of DNA that you use to contain and manage your fragment, and your donor DNA fragment, the fragment that you're interested in cloning, are combined. And they are combined to produce something called a recombinant DNA molecule. That should be the word molecule. and the organisms containing copies of the cloned DNA segment are selected. So once you put this vector and its insert into the host cell, those host cells are then grown and they are then selected to make sure that the only cells that you work with are the ones that contain the appropriate entity inside them. Some of the cells will be empty, some will contain multiple fragments that will complicate the analysis. So this is a process of filtering. Once you have amplified your cells, the next step is to extract the donor DNA from the cloning vector. And this can be done by using the same enzyme that you cut the DNA fragment with in the first place. Once you have your amplified fragments, you can do, again, a number of different things with them. One thing would be to combine them with something else to form a recombinant DNA molecule, and that can be used in any type of experiment that you desire. Incidentally, some restriction enzymes generate the same type and same length of sticky end or blunt end, and they can then be used in a complementary type of experimental fashion to generate these cohesive ends. The next step is a bit treacherous because sticky ends don't know who they're sticking to. They could be sticking to themselves by forming a circle. They could be sticking to another fragment of the DNA of interest, making two, like a train carriage. Or they could stick to the recombinant molecule backwards or forwards. So all that has to be somehow filtered away. And that's what we're saying right here. And the picture on the next slide will help us. So the techniques that have been developed is to favor just the formation of what you want and not some artifacts. So figure 15.3 is the supporting figure for the last few slides. So here's your DNA. You cut it with Echo R1. You get these fragments. Once you've spread the fragments, if you want, along a gel, you can cut these bands out, or you can use the whole lot if you want. It doesn't matter. Regardless, you have these fragments with sticky ends that are Echo R1 ends. You take your vector plasmid, you cut it with the same enzyme, and that opens up the vector. Now, in a test tube, you incubate these with these, and you can get different outcomes. You can get the vector closing back up. That's what we see here. That's no good. You can get these inserts going into this plasmid, which is what you might want. Or you can get these going in backwards into this plasmid. Or even two of these going into one plasmid. Or even two of these plasmids coming back together to form a double ring. So you can see you get lots of artifacts. The goal is to pick the one type that you're after. And that's called selection. Incidentally, the use of this term DNA ligase, hopefully you'll remember from earlier in the course. That being, whenever you want to permanently stick two DNA backbones together, you have to have this enzyme, DNA ligase. So that's what DNA ligase is doing inside the same tube as your fragments. It's putting forth the phosphodiester bond between the two overlapping sticky ends. We really don't need to go into how to isolate the clones of interest, but if you want to weed your way through these few slides, you may. And the answer lies right here in how scientists can guarantee that they are finding what they need. There's many techniques available. We mentioned plasmids. We mentioned vectors. We mentioned cloning. How do these things come together? Well, plasmids are naturally occurring extrachromosomal, that is outside the chromosome, DNA molecules. And they normally carry on them certain genes that are helpful to the host cell. Not necessary, but helpful. Vectors is simply a name for some type of entity that helps move material. So a mosquito would be a vector for malaria because it moves malaria parasite into your body. And in this case, plasmids are vectors because they help us move our DNA fragments into the bacterial cell. The plasmids are able to replicate independently of the bacterial chromosome. So they can always maintain themselves as separate entities because they're equipped with an origin of replication, an ORI. The genes that they carry may confer an advantage or give that bacterial cell some selective benefit. Or in the case of technology, they have a gene that allows that bacterial cell that contains the plasmid to be isolated from all the others that don't. So that's a form of selection. And the most common way is to use plasmids that are either naturally carrying antibiotic resistance or have been engineered to carry antibiotic resistance. Classically, the most commonly used plasmids have been puck-based, puck, and PBR-based. And there's a lot of data behind these plasmids and manipulation, and they've been very useful historically for much of the research in molecular and cellular genetics. Both natural plasmids have now been engineered into many subtypes. So there's many available from catalogs that you can purchase. For instance, the beta-lactamase gene, an enzyme, that confers resistance to ampicillin because the gene destroys the antibiotic. In addition, the origin of replication has been derived from a natural plasmid called coli-1. And the coli-1 origin of replication allows these plasmids to be maintained at a very high copy number per cell, between 1 and 200 plasmids in each cell. So that process in itself will amplify your fragment if that plasmid contains it. Now, both types of plasmids also have other regions that have been engineered, and one of them is called the multiple cloning site. It's just a small area of DNA that has been synthetically blended together to contain multiple unique restriction sites. And they are engineered so that there's only one of these sites per plasmid. So when you do cut the plasmid, it will only cut it once, as in this figure here, and it won't cut it into multiple fragments. The other thing is, in the puck-based plasmids, the multiple cloning site is within the LAC-Z gene, another gene from another bacterial operon. The system works as follows. If the gene is left intact by not having an insert, it will be able to produce an enzyme called beta-galactosidase. So this is the product of this gene. And once this protein is made, it will then cleave any chemical that it's able to identify and chemically alter. So XGAL is converted to a blue product. If and when the LAC-Z gene is broken apart by the insert, the beta-galactosidase is not made and the XGAL is not converted to a blue product, but instead the colonies remain their default color, which is white. Figure 15.6 does a great job of relaying this relationship. The general architecture of the PUC18 plasmid is indicated in the center. The location of the LAC-Z gene is also shown in orange. And within that, in yellow, we can see the multiple cloning site. Under wild-type conditions, when there's no insert, this gene is active and it produces these blue colonies. Once you put an insert into any one of these locations, the reading frame for the gene is disrupted and you get a white colony. So white colonies indicate a insert and blue colonies are bad and they indicate just a plasmid alone. Once you have your engineered plasmid, then you have the problem of getting it inside the bacterial cell. That's pretty straightforward because we rely on a natural property of bacterial cells in picking up exogenous DNA. And that process, as we know from a previous chapter, is called transformation. So the bacteria and the plasmid are incubated within the same test tube. Add in a few atoms of magnesium or electric shock the cells, and the pores on the membranes of the bacteria become activated and the plasmid is absorbed into the cell. Subsequently, the bacteria are spread out on a plate, and all you have to do then is make sure it contains XGAL, and look for those white colonies. Each colony the next day is supposed to have about 100 million cells, and each one of them should contain multiple copies of your plasmid with its insert. Therefore, you may have over 10 billion copies of your insert in a single overnight incubation. If one begins with a mixture of fragments to be cloned, then one should have a population of plasmids with inserts, recombinant molecules. Luckily for us, the process of transformation isn't that efficient. Maybe 1 in 1,000 of these cells will pick up a plasmid. The cells can then be spread on a plate and grown overnight. And not only do the cells grow, but the plasmid inside them grows, forming these colonies that are full of your amplified DNA. How much DNA can you insert into a plasmid vector? That depends on the vector itself to some degree, but there seems to be an upper limit. And the upper limit is about 20,000 base pairs of DNA. And that's hard to maintain in a high copy number plasmid. So other systems have been developed by engineers and genetic engineers, and some of these vectors are based on bacteriophages. These are viruses that infect bacteria, and they can accommodate larger inserts within their genomes. Or, as an alternative, one could use plasmids that contain low copy number inside the cell, and that helps to propagate larger fragments. If one wants to go even bigger, then we can have artificial chromosomes. Artificial bacterial chromosomes are known as BACs, bacterial artificial chromosomes, and they have an insert size ranging from 100,000 to 200,000 kilobases. And for large inserts, these are the way to go. They do contain one origin of replication, and that has been copied over from the fertility factor, a plasmid, and they contain selectable markers as well as a multiple cloning site. Sometimes your DNA of investigation may be even too large for these systems to handle. What do you do in that case? Well, you can generate cDNA libraries, or you can generate yeast artificial chromosomes, which can carry a couple of million base pairs of DNA. Let's focus in on DNA libraries. A DNA library is simply a collection of cloned fragments of DNA, usually from a single source, so from a single human being or from a single tissue within that human being. These libraries, because they're called DNA libraries, are derived from genomic DNA by default. That includes all the DNA within the cell, and this should include mitochondrial DNA if it's from an animal cell. It will also include chloroplast DNA if it's from a plant cell, and it may include neither if it's from some other organism. If, however, the library is derived from something else, for instance, just a messenger RNA produced by a particular cell at that moment in time, then those are called complementary DNA libraries, or cDNA libraries, for the following reasons. There are two major benefits of cDNA libraries over genomic libraries. Whereas genomic libraries contain all the DNA, including the introns and the regulatory sequence, the repetitive sequences, the transposons, and the non-coding intragenic regions, the junk DNA, that can complicate one's analysis and hunt for the gene of interest. cDNA libraries only represent the genes that are being transcribed at that particular time. But not only that, they also represent which fraction of the genes are being transcribed and by how much. A common example to remember in discriminating the beauty of cDNA libraries is that you could generate a cDNA library from an early liver cell in the afternoon. And then you can have liver cells that are harvested after a meal into the evening. And they may not have the same genes being transcribed at this different points in time. Therefore, the cDNA library from the early morning liver cell may not have exactly the same genes activated as the one late into the evening. And this way you can compare the change in the environment and what influence that has on genes. And the same thing with cancer cells. You could have a tumor and compare that with normal tissue in its surrounding area, and that will give you an idea of which genes are impacted in the cancer cell. Very powerful stuff. How does one construct a genomic library? Well, we've gone over many of the steps already in this video, but we'll refresh your mind. So genomic DNA is extracted from tissue, and normally a single individual, and the DNA is isolated from those cells and then fractionated into small pieces, fragmented. And we can use either restriction enzymes or some kind of sonication, sound, or even mechanical shearing of the DNA. Each one has its benefits and disadvantages. Regardless, once you have those fragments, they're ligated into cloning vectors, as we discussed in the previous section. Recombinant vectors are transformed into bacteria. Using an appropriate ratio of plasmid to insert to bacterial cell, you will end up with a huge population of clones. And if you maintain those clones and grow them on plates, giant plates, in theory you should have all the sequences from the genome present in about equal frequency amongst the selected clones. That is your genomic library. These libraries can be copied by replica plating, or they can be frozen and shared amongst different scientists. Figure 15.8 gives the rough and dirty view of how to construct a genomic DNA library. Start with your DNA, cut the DNA into fragments, clone those fragments into your vector, and then transform bacterial cells with the vector, and then select for colonies that contain the inserts, and maintain those plates, because that's your library. What distinguishes a decent genomic library from a bad one is represented here. So a good library will have a broadly representative sample of the DNA from the entire genome. It'll be large enough to be useful for sequencing and subcloning. And the inserts should be roughly of similar size, so they can be utilized in similar protocols. Construction of a cDNA library is a bit more involved, but not too much. So one thing that is present in this protocol that was absent in the genomic DNA library construction is the requirement for this enzyme, reverse transcriptase. Just keep in mind what we're looking at. We're not looking at DNA because the DNA of the cell is eliminated. We're just keeping behind a subset of the messenger RNA. Which messenger RNA are we keeping behind? Just a messenger RNA. There lies the problem. These are RNA molecules. We need DNA, double-stranded DNA. So by learning from what viruses do, we're able to get these reverse transcriptase enzymes from retroviruses and then subject our messenger RNA, once we have captured it using a system of beads with tails of oligo-DT attached to them inside columns. This is where the poly A tail of the messenger RNA will stick through complementary base pairing. Once we have recovered the messenger RNA, then we need to make a copy of it into DNA first. And then we remove the RNA and convert that copy into double-stranded DNA. That's illustrated in these two figures. So this is the cells or the tissue. In this case, we're using reticulocytes. You isolate the messenger RNA from them, and you can see all messenger RNA should have of this type some poly A tail. You put that in a column that contains these beads that contain the poly T tails. The poly T tail binds to the poly A tail and keeps that fragment of messenger RNA from being washed away. Then you add the enzyme, the reverse transcriptase, and that takes the tail of the beads and it just grows it to make a hybrid molecule. So this is DNA in blue and RNA in green. Once you've done that and you've given enough time for the enzyme to copy all your fragments, then you add another enzyme called RNase H. And RNase H simply cuts up the RNA component. And then you have to add a second primer and then get the first strand of DNA copied. And now you have double-stranded DNA, which is complementary to the original messenger RNA. To help with cloning this into your vector, you use some technology that allows you to add linkers. Linkers are simply double-stranded DNA that contain restriction sites. You add these linkers to the end, and now you're in a position to be able to clone your fragments into your vector. So your vector is in orange, and your insert is in blue. and the dark blue represent just the complementary ends of your two fragments. And this plasmid happens to contain an ampicillin resistance gene, which allows it to be selected, and also an origin of replication. As we mentioned briefly earlier, the benefit of these libraries is that they're tissue specific. And not only that, they also lack regulatory sequences and all the other extraneous DNA components, including introns. So cDNA libraries are valuable because they only represent what that cell is making in terms of messenger RNA at that time. Additional benefits include the level of representation of different messenger RNA types is proportional to the activity of that gene, i.e. how many messenger RNAs was it encoding relative to the others. So in this particular example, we see a comparison of genomic versus two types of cDNA library. So genomic library would take all these fragments, irrespective of how much they are being expressed, and make us a representative library. So each gene is represented approximately the same as the others. In a cDNA library that comes from the embryo, in the embryo, gene 2 and gene 3 are being expressed at maybe different levels, and that's represented here. So gene 2 has 2, 4, 6 clones for every 2 clones for gene 3. That tells us in the embryo, gene 2 is more active than gene 3. But if we were to wait and then make the library from the eyes and compare them, you would find that in the eye, gene 1 is much more active than gene 3, but gene 2 is no longer active, because gene 2 is only active in the wings. Once these libraries come into existence, then they can be used in a multitude of different ways, including sequencing and the creation of reporter genes. If one is involved in reverse genetics, the manufacture of artificial or mutated alleles of genes is a very important tool in your investigation. Current limits on technology allow us to make up to 50,000 base pairs of DNA at will using PCR-based technologies. In the future, that level may be extended many magnitudes further. In fact, there may be a point in the near future where we can make an entire chromosome at will, and even an entire genome. The second section of Chapter 15 mentions the technologies available for taking genes from one creature and putting them in the DNA of another. These would then be considered transgenic organisms, and the manipulated gene would be called a transgene. Care must be taken with these technologies because the transgene first has to be integrated into the genome, a process known as integration, and then you have to be able to turn that gene on and off as you expect using appropriate regulatory sequences in the right places around that gene. Luckily for scientists, almost every creature or every genome utilizes the same genetic code for controlling RNA and for making proteins. However, the regulatory parts are handled differently in different branches of the tree of life, and these have to be then suitable to the gene, the transgene, that one is transferring into the host. For something to be considered a transgene, the DNA has to come from another organism. If you are manipulating the organism's own DNA, then that is not considered a transgenic experiment. Using our basic knowledge of genetics and biology, scientists in the past have engineered the plasmids and the vectors to carry the appropriate regions in the right places in order to transcribe the transgenes. That involves having expression vectors fully furnished with upstream promoters and multiple cloning sites that also contain the Shine-Delgado sequence needed for the ribosome to bind to the messenger RNA and translate that into protein. One thing that bacteria cannot handle are introns. They don't have a spliceosome able to splice out introns. Thus, any transgenes have to be coded correctly or made from messenger RNA. Figure 1511 shows us a prototypical expression vector used in E. coli. Please take a moment and look at each element in this drawing and see if you can relate to its purpose in the construct. If the appropriate promoter or enhancer regions are included, then we can induce the expression of the gene in the host by using inducer molecules or inducer compounds, or we can have it expressed all the time constitutively. In one classic setup, we are able to fuse the LAC operator, the cap binding site, and the RNA binding site to control the activity of the transgene in the same manner as we would the natural LAC operon using lactose and glucose. Moving on now to look at eukaryotic expression vectors. These have in the past been restricted to either yeast cells, a type of fungus, or to tissue culture cells made from the tissue of the organism that you're investigating. The reason for switching away from bacteria is that the bacterial cell doesn't contain the appropriate machinery to interact with that DNA in a suitable manner. These eukaryotic expression vectors have to still abide by the same rules as bacterial expression vectors, namely, they must have all the necessary elements for the eukaryotic system, the cell, to regulate, transcribe, and translate. The textbook uses the example of human insulin production in E. coli bacteria. So the gene for human insulin was one of the first to be isolated and then tested inside bacteria in these genetic engineering experiments in the early 1980s. Insulin itself is not the product of the gene. The gene first produces a 110 amino acid protein called a pre-proinsulin, and that's made inside an organism, inside the pancreatic cells. And that is not secreted and does not function until it's processed within the cell. And within the cell, after production, the first 24 amino acids are removed, forming proinsulin. Then a further 35 amino acids are removed. But we haven't finished yet. So after the first two cuttings of the initial polypeptide, additional cleavage generates two amino acid chains, the A chain and the B chain. And these are held together by disulfide bonds. And that's what insulin is. It's not a single polypeptide, it's two polypeptides which came from the original parent polypeptide that was itself processed. So getting all this correct inside a bacteria may be difficult, but a pancreatic cell can do this. Figure 15.12 illustrates this relationship between the amino acid sequence of the human insulin B chain that was determined by peptide sequencing and a nucleotide sequence that was created by reverse translation of the amino acid sequence. That was the technology available to us in the latter half of the 20th century, before DNA sequencing was invented. In order to enable bacteria to make human insulin, a number of significant manipulations were necessary, including the construction of fusion genes so that fusion proteins could be produced. In this case, the fusion gene was the reporter LAC-Z gene fused to either the A or the B chain DNA sequence. In addition, codons for methionine were added so that translation would ensue on the ribosomes of the bacteria. The vector construct for the B gene is indicated in figure 15.12, and the reading frame has to be maintained so that the fusion protein can be made correctly. Please remember that this construct was made chemically without using DNA synthesizers. This happened in the late 1970s. It was a remarkable feat. The company Genentech was one of the players in this technology. Once the bacterial cell transcribed the information on the plasmid, The translation process would continue to produce a fusion protein. Fusion proteins will not work in a human subject. Therefore, an alternative method was needed in order to cleave the LEC-Z away from the A or B peptide of insulin. Cyanogen bromide treatment is able to cleave the C-terminal end of methionine, and that was then used to separate the fusion protein into two halves. Additionally, the use of lactose as the inducer and putting the bacterial cells in a medium without glucose was the key in regulating the activity of this gene. If you're not familiar with this system, you may want to go and visit the chapter on lactoperols. Following cyanogen bromide treatment, the A and B chains are purified from the host strains by lysing the cells and then mixed together under conditions that allow the disulfide bridge to be formed. And now you have human insulin that can be sold to your customers. Figure 15.12 just shows you the final leg of that technological marvel. The employment of bacterial workhorses for human benefit goes beyond simply human insulin. The historical record shows that bacteria have helped humans in the manufacture of human growth hormone, erythropoietin, a hormone for red blood cell manufacture, even laundry detergent additives that help stain removal. The list goes on and on. One industrial application of gene technology that we are all impacted by is the production of cheese. So engineering both bacteria and fungi to produce the enzyme chymosin, which curdles milk. The manipulation of yeast vectors and host cells can, in most cases, overlap the technologies used with bacterial systems. Natural strains of yeast harbor a circular plasmid called 2 micron. This plasmid has been manipulated to include an E. coli origin of replication and suitable markers and cloning sites, therefore enabling it to be a vector. Now, because the plasmid can be moved between bacteria and yeast quite easily, it's called a shuttle vector. A shuttle vector is a cross-species, cross-domain entity, which can be used quite easily in either system because it has all the necessary components to satisfy both classes of cells. We mustn't forget plants. Plants are an enormous component of our life cycle. How about plant manipulation? Well, naturally, there's a relationship between agrobacterium and plants. The agrobacterium is the bacteria that lives in soil, and it fixes nitrogen for most of the plants in the world. So this is a relationship that's evolved over many hundreds of millions of years. In natural conditions, the bacterium has come up with some tricks in order to get the plant to supply the bacteria with what they need. One such trick is to use a plasmid known as Ti to cause the cells of the plant in close association with the bacteria to be transformed and divide rapidly, forming root tumors. These root tumors can extend above the soil, and at that point they're known as crown gall disease. The entire plasmid is known as Ti plasmid for tumor inducing, and the part of the plasmid that's given over to the plant cell to make it misbehave is called the transfer DNA or tDNA. This was mentioned in another chapter of this textbook in a different context. Once inside the plant cell, this small fragment of DNA, the tDNA, can undergo illegitimate recombination to integrate into random locations within the plant genome. Within the tDNA sequence are genes that cause plant cells to divide uncontrollably and produce amino acids that are unique to the bacteria. These amino acids are not your standard 20 amino acids. These are known as opines. and only the bacteria can use them as a source of energy, which then diffuse across the plant cells to feed the bacteria. The genes inside the tDNA resemble, to some degree, the fertility factor found in the plasmid of E. coli. Figure 1513a shows us the arrangement of the plasmid, including the T region that's responsible for causing crown gall disease. The bottom half of the figure shows us an example of one of these tumors on the side of a tree, as well as the mechanical transfer from the bacterium to the plant cell. Scientists have harvested this natural system by replacing some genes in the tDNA with the genes of interest. So the vector still is able to infect the cells of plants, but it doesn't cause undifferentiated growth. Instead, it introduces a gene into the genome of the plant. As long as one's construct stays within the boundaries of the tDNA, the natural system will transfer your transgene into the host genome. As with bacterial and yeast systems, the inclusion of selectable markers makes it easy to determine which experiments have succeeded and which have not in a form of screening. The commonly used screening genes are either for herbicide or an antibiotic that allows those cells to be selected. In a further evolution of the technology, many newer experiments use a disarmed Ti plasmid and a second plasmid containing only the left and right border flanking sequences of the gene of interest, as indicated in figure 1514a. So take your time and try to understand the content of this slide in relationship to the past 3 or 4 slides. In order to investigate the competency of your experiment, you have to grow the transformed plant cells into an entire plant in order to see the effects of the experimental outcomes. Luckily for us, most plant cells are totipotent. That means they contain all the information necessary to form a complete plant, and therefore they can be regenerated from these transformed cells. The second half of figure 1514 relays these concepts. So here are your 2 plasmids. This is the transfer taking place to the plant cell, and then the subsequent incorporation into the genome of the plant. Part C just shows us how cultured cells can be then grown into entire plants that are transgenic. 2 of the most manipulated traits when it comes to plant transgenics are the resistance to herbicide and the resistance to insects. So herbicide-resistant crops allow farmers to treat entire fields with that herbicide, for instance Roundup, and the Roundup will kill the weeds, but it won't kill the crop plants because they contain enzymes able to neutralize the Roundup and other herbicides. Likewise, insect resistance can be conferred by tapping into a natural bacterium, Bacillus thuringiensis, which produces a toxin called Bt. And if you express that toxin in the plant, it acts as a natural insecticide. A commonly cited example of the use of technology to manipulate plants for the good of humanity is golden rice. So rice itself, wild-type rice, is a major staple for much of the world's population. The most commonly consumed part of the rice is the grain itself, the endosperm. However, that lacks some important micronutrients, including provitamin A, also known as a carotenoid. This substance is converted in the consumer's body to vitamin A. It's important to realize that this is lacking in the endosperm of normal rice strains. The husk of the seed grain does produce provitamin A. So it's not that it's not made in the part that most people eat. And if you read the textbook, it tells you the reason that they remove the husk is that the husk contains oils that become rancid with age and the added heat of the tropics. What scientists learned is that the endosperm does synthesize a chemical, granyl diphosphate, GGPP or GGPP, and that is a precursor to the synthesis of these carotenoids, which can then be converted to vitamin A inside the consumer. Here's a diagram showing you how evolution has taken GGPP, and in most plant tissue it's able to convert that into a beta carotenoid. Bacteria can also take this GGPP and through a slightly different pathway also convert it to beta carotenoid. So the scientists looked at both pathways and they realized that these three enzymes could be easily substituted by this single enzyme from bacteria. Combining this knowledge with the findings of other scientists in 2000 revealed that by adding just 2 genes, these rice plants could enable them to convert GGPP into beta carotene. The second gene was derived from a daffodil plant. This produced a new variety of rice known as golden rice. Golden rice has gone through a couple of iterations. This is white rice, and you can tell by the color of the grains that it contains no beta carotene. This is the first generation of golden rice, GR1, and that was generated through the use of the daffodil and the bacterial enzymes. Since then, an updated, revised strain of golden rice has materialized, and that uses a gene from maize instead of from the daffodil. This swap enabled more beta carotene to be produced inside the rice grains, and we can see this as a darker pigmentation in GR2. Further work has continued on this project with the following objectives. First, the transgenes should only be expressed in the endosperm and only during endosperm development, no other time. Second, the beta carotene synthesis should be increased by using different versions of the genes to suit different environments and different cultural tastes. And the selectable marker should be removed from the transgenic lines by some mechanism. And finally, these transgenes could be introduced to strains used by subsistence farmers in parts of Africa and Asia. So those strains of rice are different to the ones that the golden rice project engineered. Transgenic plants, like any other type of transgenic organism, have raised moral, ethical, and legal concerns. Some people fear that human health may be affected by an increase in allergic reactions and asthma, and others fear the escape of these transgenes into the wild populations, especially our food plants. Just to finish off this section of the chapter, we're going to turn our attention very briefly to look at transgenic animals. The problem with transgenic animals is that homologous recombination occurs less frequently than does illegitimate recombination. And the problem with illegitimate recombination is that it can recombine with parts of the chromosome that may be critical to the survival of the animal. The other thing is, totipotency is not a general characteristic of animal cells. Only certain cells in the developing embryo are totipotent. The manipulation of these types of totipotent cells, which includes embryonic stem cells, leads to its own problems and concerns because you're dealing with genetic mosaics. With respect to animals, the research on vertebrates has come a long way, and the mouse has been the champion of transgenic studies. A general approach to creating transgenic vertebrates involves injecting DNA directly into the nucleus of a fertilized egg cell, one that has been fused with a sperm. This injected DNA then integrates by the illegitimate recombination process into random locations in the genome. Therefore, each individual transgenic animal will have a transgene in a different genetic location. In addition, there are two other factors that must be taken into consideration. The first is the production of concatemers. Concatemers are simply molecules that are repeated one after the other, like links in a chain. And we call these multi-copy concatemers. They often result in the incorporation into the chromosomes, and the fact that there are multiple copies leads to the abnormal expression of those genes. And the second concern is that expression of the transgene may be abnormal due to the location of the chromosome in which it sits. If it sits in the highly active area, it may be expressed at a higher level than if the gene was to be inserted into a silent area of the chromosome. One of the success stories, maybe, is the generation of transgenic salmon. So these are wild type salmon, and these are the transgenic cousins produced by genetic manipulation. The difference is that the promoter region of the wild type salmon is sensitive to light, and that light is only available at the intensity during spring and summer in the environment in which these fish normally live. What the scientists did is they replaced this segment with a segment from another gene that is not responsive to light. It's on all the time. So this enhancer is going to keep the Tata box and the promoter region active all year long. So these fish continue to grow full time throughout the entire year. By using transgenic mice over the last 30-40 years, scientists have been able to dissect many aspects of human development and physiology, as well as apply the knowledge back into mouse development and physiology. Newer methods are being developed all the time, but the most important advance has taken place with CRISPR-Cas9 gene editing. This has revolutionized our need to even use these types of animals. When it comes to recombination, there's two types. There's homologous recombination and there's illegitimate recombination. In order to discriminate between successful outcomes and unsuccessful outcomes, scientists have developed two forms of selectable marker. One is called a positive selectable marker, that being, in most cases, the neomycin gene. Neomycin gene produces a product which metabolizes the drug G418, which is normally lethal to mammalian cells. So these cells are grown in a dish or culture that contains G418. Only those cells that have been transformed correctly and therefore express the neomycin gene that's carried by the transformation vector will be able to survive and grow. In addition, in order to be extra careful, scientists started including a negative selectable marker also. This gene was placed outside the region normally involved with homologous recombination. So if illegitimate recombination took place, then this gene will be transferred onto the chromosome of the transformed cells. A very commonly used negative selectable marker is thymidine kinase, or TK, and that comes from the herpes simplex virus. The product of this gene interferes with DNA synthesis when cells are grown on a plate containing thymidine analogs, GAN cyclovir. The logic is straightforward. If homologous recombination takes place, only the positively selectable marker will move onto the chromosome. If illegitimate recombination takes place, then both genes will move onto the chromosome. And because the second gene is lethal, those cells will not survive. This system is known as positive-negative selection, because both of them can take place at the same time. Figure 1518a does a great job of explaining the architecture. So if you follow pathway 1, this is the desired pathway. This is what you expect the experiment to generate. Here the recombination is homologous, so the blue areas are compatible with the chromosome and the vector, and then the region in between is transferred over to the chromosome. And those cells will have a positive selectable marker only. In the case of illegitimate recombination, recombination will take place outside the dark blue areas in the light blue areas, and that will go in anywhere on these chromosomes. But the transfer of the second gene is also encompassed within the crossovers. And these chromosomes will contain the thymidine kinase, the negative selectable marker, and those cells will be killed in the presence of the chemical when they try to reproduce. Let's talk further about embryonic stem cells. Embryonic stem cells are unique in that they are equipped with the ability to produce another human being with everything needed, including the placenta. So those type of cells are known as totipotent. Where do you find embryonic stem cells? Embryonic stem cells are only found at a certain stage in the blastocyst. So this is the blastocyst. These cells will make the embryo. These cells here will produce the placenta. But they all came from a fertilized egg, all these cells. In a standard protocol, once the embryonic stem cells have been harvested from the blastocyst, they can be grown almost indefinitely in culture. At that time, DNA is introduced into these cells, so the cells become transformed, and then the cells are transferred to a media that selects for the positive marker and against the negative marker. Once confirmation of the identity of the genetic manipulation has taken place, the transformed cells are reintroduced back into a blastocyst, but maybe into one from a genetically distinct creature. Therefore, you can identify the two populations of cells. The blastocyst is implanted into a surrogate female, and the mouse that's born will be a mosaic in nature. If that mouse survives to adulthood, it will also produce its own gametes, some of which will contain the chromosomes from the transgenic. So the offspring produced by these will be heterozygous for the transgene. Figure 1518 takes one through a journey of such an experimental manipulation. In this particular example, the CFTR, the cystic fibrosis transmembrane conductance regulator, a membrane protein whose non-function leads to cystic fibrosis, is studied. The goal is to generate these homozygous knockout mice where both copies of the transmembrane regulator are mutated. See if you can follow the logic. There's nothing new here that you don't already know about Mendelian genetics. In some experimental designs, it's necessary to manipulate the transgenes, not in the Petri dish, but inside the organism. And there are methods available to do that too, but they're pretty complex and outside the scope of this lecture. Let it just be said that by using what we just learned in the previous section about positive selection and negative selection, and how things can be added and removed from genes by recombination, these are the techniques that have been then taken to the next level. So one of these is called site-specific recombination, and that requires some bacteriophage biology. If you wish to follow through, then the next 6 slides are designed to take you through the logic behind how that system works. It's just alternative types of recombination that can be then manipulated by the scientist. Third and fourth sections of the chapter are pretty brief. because they refer students to other chapters to get the detail. But gene therapy uses recombinant DNA technology, and in that regard we're going to be looking at mostly the CRISPR-Cas9 system. The overarching goal of this application of technology is to cure or at least alleviate some of the symptoms of diseases. If this can be done, then we are performing gene therapy. The goal is to be as precise as possible when changing things. And as we mentioned, the CRISPR-Cas9 system gives us a very powerful tool to abide by this rule. Gene therapy itself is classified into two categories. There's somatic gene therapy and germline gene therapy. The difference is straightforward. Somatic gene therapy targets somatic cells that will never give rise to germ cells. So any manipulations performed in that organism will stay within the organism and not be passed on to its offspring. On the contrary, germline gene therapy targets germ cells, and any manipulations performed there will be passed on to the offspring of the treated individual. Now, for ethical reasons, the scientific community had a moratorium on using germinal gene therapy. However, recently, scientists in China has broken the mold and has produced 2 human offspring, with a third one pending, by manipulating germline cells. Some tissues of the human body are more amenable to gene therapy. For instance, liquid tissue like blood and lymph can easily be extracted from the patient, treated, and then reintroduced back. However, for other disorders, such as for solid tissue, cystic fibrosis included, it's hard to remove that tissue and treat it outside and then return it. The latest renditions of gene technology enable embryonic stem cells to be manipulated by CRISPR-Cas9 systems, therefore producing customized cells that can then be used to initiate pregnancy. One of the major hurdles overcome by scientific research was the difference between programmed and unprogrammed cells. Those are known formally as differentiated and undifferentiated cells. Embryonic stem cells would be classified as undifferentiated cells, and adult cells would be classified as differentiated cells. The goal was to reprogram the DNA so that it would think that it's part of an embryonic undifferentiated cell. Everybody should know that all cells in your body, with the exception of red blood cells, contain the entire genome. So why doesn't a skin cell want to go back in its life cycle and behave like an embryonic stem cell? Well, the scientists were able to use just 4 transcription factors to reprogram cells. And these factors are named after the scientist Yamanaka, who first discovered these 4 transcription factors. So these 4 transcription factors are like master genes. Their activity produces 4 proteins, and those 4 proteins are able to manipulate the genome of a cell by epistatic and other mechanisms and reinitiate the programs of genetic control to take this cell back to a life of an undifferentiated cell. The latest research has shown that these transcription factors work in combination with proteins known as pioneer factors. These are the ones that can open up heterochromatin into euchromatin and reprogram that at the epigenetic level. Recently, viruses have been used as delivery vehicles, particularly those viruses which target specific tissues around the body. These viruses have to be disabled, and that's done easily by removing certain genes, and the space created by the removal of those genes allows us to introduce our own genetic material, such as the genes to correct cystic fibrosis, and by spraying these adenoviruses into the nose of the patient, there's a temporary relief from the disease, at least in the nasal cavity. The viral techniques have their challenges, as do the induced pluripotent stem cell techniques. In the case of viruses, their integration into the host's genome may manifest in detrimental outcomes. With induced pluripotent stem cells, they continue to express these transcription factors, and that predisposes the cells in patients that have been treated with this form of therapy to tumors and cancers. There are some beautiful success stories in the literature. One of them is the curing of sickle cell disease in mice. The strategy is pretty straightforward. Mice adult cells are harvested. They are then reprogrammed into iPS cells using the transcription factors. The genetic defect is repaired. These cells are differentiated into blood cells and then transplanted back into the bone marrow of the affected mice. Once the research team had a proof of concept and the results were clarified by their peers, they moved on to look at humanizing the mouse model. What they did was to take out the mouse alpha globin gene and substitute that with the human alpha globin gene. And they were able to remove the mouse beta globin gene and replace that with the human beta globin sickle allele gene. Mouse fibroblast cells were collected from the tails of these homozygous mice and infected with retroviruses encoding the genes for 3 of the Yamanaka factors, the transcription factors. And the 4th gene came from an lentivirus. Expression of the Yamanaka factors resulted in reprogramming of the fibroblast cells into iPS cells. Using knowledge from the LOX and Cree systems, the c-Myc gene was excised from the mouse genome, and homologous recombination was used to enforce gene replacement. Today we could do the same complicated experiment by simply employing CRISPR-Cas9, whereby in this 1, 2, 3 system, the linear DNA template carrying the wild type version of the gene, number 3, will be injected into iPS cells along with the CRISPR, number 1, and the guide RNA, number 2, components. And through homologous recombination, once the CRISPR system had cut within the gene, the insertion of the normal copy of the allele would take place. Using either the classical method or the new Cas9 system, the results were that the mice were cured of sickle cell disease. As always, the concerns exist with various steps of these protocols. Returning back to the retroviruses that have been used in some experiments, they can cause unintentional mutations, and because they either carry oncogenes or they insert themselves and activate oncogenes, the potential to cause cancer in the subjects is of great concern. With iPS cells, the major concern is, are they really totipotent? Can they give rise to all the tissues of a body? The technical challenges permeate into the types of diseases and the tissue that they impact. The list of success stories involving CRISPR-Cas9 continues. Another major disease of humans is Duchenne muscular dystrophy. It's a progressive weakening of the muscles caused by loss of function and eventually death. The biology of this particular disease makes it particularly amenable to Cas9 treatment. The last part of the chapter looks at cloning of entire animals or plants. In these processes, we bypass the natural sexual reproductive mechanisms of animals, and we rely on the asexual propagation capacities of plants. Because asexual reproduction does not involve a second parent, the genes do not get segregated. Therefore, any beneficial traits will be passed on to the offspring directly. Most of us are familiar with this process because we or our parents have taken cuttings of plants and used those cuttings to regrow an entire organism. With cloning of animals, animals do not really propagate by asexual means, although there are some exceptions. such as some species of aphids. With most animals, the totipotency of embryonic stem cells is just limited to that period of time in which those embryonic stem cells are differentiating. After that point, they become pluripotent. Combine that with the ethical and moral challenges of working on embryos leads to complicated outcomes. One universally touted success story has to do with Dolly the sheep. Dolly was a sheep. She was the first cloned mammal in recorded history. Her origins began by a scientist removing the diploid nucleus from an adult cell and then using that nucleus to inject into an egg that had its own nucleus removed. Then shocking that egg using electric current, initiating development, and then transplanting the developing embryo into a surrogate animal. Dolly did live a normal, healthy life, but it took 270 tries before Dolly was actually confirmed as a pregnancy. The final figure, figure 1522, relays the scientific process. So cells were removed from this donor sheep. There they are. The nuclei were taken from these cells, and the rest of the cell was discarded. At the same time, another species of sheep was used to donate an egg, an unfertilized egg. Its nucleus was extracted using a similar technique, and the nucleus of the first sheep, the fin dorsal, was injected into this egg. After electroshock, the egg was implanted into a surrogate mother after it developed into the blastocyst stage. This mother had nothing to do with the genotype of either the egg cytoplasm or the nucleus. But Dolly was subsequently born. And there's Dolly with her surrogate mother. Dolly went on to have babies of her own, and then she finally succumbed at a nice healthy age. Today, the cloning of dogs and other personal pets is standard, and for about $300, we can go to South Korea and have your dog cloned. Thank you for listening.